Good afternoon all. So in the yesterday's class we have discussed about the anaerobic culture methods, and we have discussed the brief introduction about the anaerobic culture methods, and also we have started the use of different methods. One of them is vacuum desiccator we have discussed. Then we have discussed about the Macintosh jar. So before describing the further other methods, I think I want to describe some more important points concerning the anaerobic culture methods. That is, what are the different classification of microorganisms on the basis of their relationship to oxygen and carbon dioxide. So first of all, coming through these points, then we can discuss different methods. Already I have discussed Macintosh filled jar, candle jar, and also vacuum desiccator. And after describing this, we can discuss rest part of the culture methods. Okay. So obligate aerobes, according to the category, that is 15% to 21% oxygen is required for them. An example for this obligate aerobes are mycobacteria and fungi. Next is microaerophil. They require 5% oxygen. Example, Campylobacter and Helicobacter. Then coming are facultative anaerobes. They multiply equally well in the presence or absence of oxygen. And the most important group of microbes coming under facultative anaerobes are Enterobacteriaceae and most of the staphylococci. Then coming is aerotolerant anaerobe. They require a reduced concentration of oxygen, that is anaerobic system and microaerophilic environment. And examples for this group are most strains of streptococci, propionibacterium, lactobacillus, and some clostridium. Next category are obligate anaerobes. They are strict anaerobic. They require strict anaerobic environment. That means 0% oxygen. Most of the bacteroid species, many clostridium, eubacterium, fusobacterium, peptostreptococcus, and porphyromonas. So these are true anaerobes. Next category is capnophil, which means carbon dioxide loving. They require 5% to 10% carbon dioxide. Some anaerobes and Neisseria comes under this category. So these are the classification of microorganisms on the basis of their relationship to oxygen and carbon dioxide. So why this oxygen is toxic to anaerobes? Because enzymes superoxide dismutase and catalase are absent in anaerobes. So, and these enzymes convert lethal hydrogen peroxide or superoxide anion formed in their cells due to the presence of oxygen. So because of this absence of this enzyme, this lethal hydrogen peroxide is not converted. So what happens? This is very toxic and they may kill the anaerobes. That means oxygen plus electron, which gives superoxide anion. And this oxygen plus electron plus 2H plus gives hydrogen peroxide. Then 4 oxygen plus 4H plus gives superoxide dismutase and gives 2 water molecules and oxygen. And these 2 water molecules, in the presence of catalase, they are converted into 2 hydrogen peroxide molecule. In the presence of the enzyme catalase, they are converted to 2 molecules of water and oxygen. So that's the reason why the oxygen is toxic to anaerobes. Now, these are the some of the endogenous anaerobes of various anatomic sites. That is skin. In the case of the skin, the most important anaerobes are propionibacterium, peptostreptococci, and in the upper respiratory tract, the most important anaerobes are peptostreptococci actinomyces. Then propionibacterium, campylobacter, fusobacterium, prevotella and veillonella. In the oral cavity, the most significant anaerobes are actinomyces, eubacterium, peptostreptococci, campylobacter, fusobacterium, prevotella, bifidobacterium, porphyromonas and veillonella. In the case of intestine, the most significant anaerobes are bifidobacterium, eubacterium, egertella, clostridium, peptostreptococci, Bacteroides fragilis group, and Bilophilia campylobacter. And in the case of the genitourinary tract, most important anaerobes which are significant are Peptostreptococci, Bifidobacterium, Fusobacterium, etc. So these are the endogenous anaerobes of various anatomic sites. So these are the sites where the anaerobic infection occurs. So these are the list of the infections caused by anaerobes. So you can check through this, that is actinomycosis, antibiotic associated diarrhea, brain abscess, infection of female genitourinary tract, intra-abdominal infections, liver abscess, peritonitis, myonecrosis, oral sinus dental infections, aspiration pneumonia, and pleuropulmonary infections. So these are caused by various anaerobes. Next is the specimen selection and collection of anaerobic culture. The specimens usually are collected from warm, moist environment that is low in oxygen. And the specimen collected by needle and syringe are better for anaerobic bacteriology than swab because they are less likely to be contaminated by endogenous anaerobes. Following the aspiration of specimen, any air present in the syringe and needle should be rapidly expelled. Otherwise, it may move towards other bacteria. It must be rapid transport to laboratory and processed rapidly with minimum exposure to oxygen. So these are the acceptable specimens of anaerobic bacteriology. In the case of central nervous system, acceptable specimens are CSF, aspirated abscess, tissue from biopsy or autopsy. In the case of the dental, ear, nose, throat specimens, aspirated abscess material, biopsy tissues are required. Next, in the case of localized abscess, needle and syringe aspirations of closed abscess. Mainly, these aspirated materials are collected from each of the sites. And swab is not recommended, it is discouraged. But under certain conditions, they use swab also. So those are the unacceptable specimens for anaerobic bacteriology. Throat swabs, nasopharyngeal swabs, sputum obtained by nasotracheal or endotracheal suction, bronchial washings or other specimens obtained through a bronchoscope, expectorated sputum. Next is gingival swabs or any other intraoral surface swabs, fecal rectal swabs, gastric contents, voided or catheterized urine, vaginal, cervical or urethral swabs, and surface swabs from decubitus ulcers. These are the unacceptable specimens for anaerobic bacteriology. Why they are not they are unaccepted? Because there is a chance of contamination with aerobes. Next is transport of specimens. Different kinds of anaerobic transport system are available. One is rubber stoppered collection vial containing an agar and indicator system. That is, the vial is gassed out with oxygen-free carbon dioxide or nitrogen. Specimen is injected through the rubber stopper. After all, air is expelled from the syringe and needle. For only a swab specimen, a special collection device with an oxygen-free atmosphere is required. When reinserting the swab, care must be taken not to enter any air. Next is anaerobic bags or pouches. Tissues can be placed in a small amount of liquid saline to keep it from drying out and placed in an anaerobic pouch. So, these bags are useful as transport device. For example, biopsy specimen. All the specimens should be held at room temperature. Refrigeration can oxygenate the specimens. Any specimen with suspected anaerobic bacterial infection must be sent to the laboratory into anaerobic transport medium or under anaerobic conditions such as cooked meat broth or Carey-Blair medium. The aspirated material into the syringe for anaerobic culture may be simply sent to the lab by bending the needle or probing the needle into the rubber cork. So what happens in this case? The aspirated materials into the syringe for anaerobic culture may be simply sent to the lab by bending the needle into the rubber cork to prevent the specimen from oxygen contact. So these are all performing to prevent the contact with oxygen. Next is the processing of specimen. That is macroscopic examination of specimen, preparation of gram stain smear for microscopic examination, inoculation of appropriate plated and tubing media. Then anaerobic incubation of inoculated media. So these are the different types of processing methods of the clinical specimens which are suspected with anaerobic infections. So up to this we have covered the portion. The next part we can discuss later. Thank you.